So I'm going to concentrate my remarks this afternoon on attempts to develop T-cell therapies for patients with the solid epithelial cancers. But I want to just take a moment to go back a little bit in history because we face an interesting anniversary in the next couple of weeks. And just talk about that for just a moment. I don't have a disclosure slide. I work for the U.S. government, and so unhappily I have no personal disclosures to show you. Can I have the next, can I show the slides?
Yeah.
Here we go. Okay. So I pulled this piece of paper out of my file when somebody pointed out to me the event occurring in about 3 weeks. This is a review of the very first human gene transfer protocol that I kept in an ongoing fashion when we were first applying for the first permission to give foreign genes to patients. We had performed studies showing that we could use T cells to cause the regression of patients with melanoma. We can now do that in 30% of all patients with melanoma by adoptively transferring their own T cells. But when we first proposed to insert foreign genes into these lymphocytes and give them, there was a huge uproar of review. We went through 16 different review groups. I was working very closely at that time with French Anderson and Michael Blaze, who are in the National Heart, Lung, and Blood Institute and the Human Genome Institute, and the three of us put together a clinical protocol to put foreign genes into a patient's lymphocytes, neomycin phosphotransferase genes, so we could track where they went. We went through these 16 review groups and finally got to the Recombinant DNA Advisory Committee that after much discussion approved our ability to give these gene-modified lymphocytes. This has never been, no patient had gotten gene-modified cells of any kind. The Recombinant DNA Advisory Committee voted 16-5 to allow us to do it, but James Weingarten, then the director of the NIH, said, no, this has got to be a unanimous decision by the RAC before we could proceed. So we went back and forth with each of the review groups, and finally, the Recombinant DNA Advisory Committee voted 13-0 with one abstention to enable us to proceed, and no sooner did we get that permission at the RAC meeting than Jeremy Rifkin, a biotechnology activist, filed a lawsuit against the NIH, bringing disabled people to the RAC meeting, claiming that it was not ethical to tamper with the human genome. That then delayed things for another 9 months until finally, on May 22nd, 1989, we received permission and actually treated the very first patient with their own T cells that were transduced with a gene for neomycin, retrovirally transduced with a gene for neomycin phosphotransferase. So in another 3 weeks is the 30th anniversary of the first time that any genes were inserted into patients. Of course, these were not therapeutic genes. We published that in 1990, and there are many people in the audience. I've already run into several walking around this morning, just coming here, who are part of this adventure back in 1989 and 1990. So here we are now, 30 years later. In 2017, as you know, the first gene therapies with CAR T cells were approved for patients with lymphomas. We concentrated on patients with diffuse large B-cell lymphomas, the most aggressive form of lymphoma to treat with CAR T cells. And in fact, these can be very durable. We treated 28 patients. 50% of them had CRs, and of those 12, all but one have ongoing CRs. With median follow-up beyond 4 years. The first patient we ever treated with lymphoma we treated in 2009, who is in an ongoing complete response now, almost 10 years later. But that's the lymphomas. This is, however, on this slide, is the problem of cancer in the United States today. According to the American Cancer Society, according to the American Cancer Society, okay, good, thanks. According to the American Cancer Society, there were 600,000 deaths in Americans due to cancer last year. 1 in every 2 to 3 Americans now alive will develop an invasive cancer, and 1 out of every 4 or 5 of us are going to die of a cancer unless we find better treatments for this problem. Now notice that the hematologic diseases for which CAR T cells are showing increasing importance, not only in lymphomas but in multiple myeloma, they comprise about 10% of all cancer deaths. Over 90% of all cancer deaths are due to the solid epithelial cancers. These are the organs that have ducts, that have epithelial linings, the colon, the stomach, the pancreas, the ovary, the esophagus. It's those cells lining the epithelial lining of the ducts that are dividing, make mistakes in their DNA division, create mutations, and that results in cancer. And it's those solid cancers that killed, again, 90% of everyone who dies of cancer. And so the major challenge confronting modern cancer immunotherapy is the development of effective immunotherapies for patients with these metastatic solid cancers that cannot be cured by any available treatment and result in the majority of cancer deaths. Now I'm going to concentrate on cell transfer therapy. We first used this in 1988 to show that lymphocytes were the, in fact, final mediators of anti-tumor responses in patients with melanoma. And so there are many advantages of cell transfer immunotherapy. You can grow well over 10 to the 11 lymphocytes quite easily over the course of several weeks. If you can identify cells that can recognize the tumor, you can activate them ex vivo so that they're not subjected to the anergic and tolerizing effects that exist in vivo. Because you have the cells in the test tube, you can identify the exact cell subpopulations and effector mechanisms that are responsible for tumor regression. And most importantly, because you've removed the immunologic reagent from the patient, the T cells, and you're going now to manipulate or find them or genetically modify them, you can irradiate the host, lymphodeplete the host to eliminate the inhibitory mechanisms that exist, T regulatory cells, myeloid-derived suppressor cells, intracellular cells infiltrating into the stroma of tumors that produce inhibitory cytokines. And it's this ability to manipulate the microenvironment of the tumor in patients that makes cell transfer therapy so important. Now, we've heard a lot about CAR T cells. And I personally feel that right now there is no application for CAR T cells for the solid tumors in adults. And why is that? Well, we learned some the hard way. CAR T cells require the use of monoclonal antibodies. You've heard the difference between CAR T cells and a conventional T cell receptor. You have to have a monoclonal antibody and intracellular signaling chains that will cause that lymphocyte to signal. Well, they were first described over 43 years ago by Kohler and Milstein, and despite some of the best minds in the field, no one has ever found a monoclonal antibody that is unique for cancer that isn't also recognizing normal cells in the adult. And therefore, CAR T cells that will recognize a tumor will often recognize the cell the tumor came from. And in fact, we've learned the hard way that these T cell receptors and CAR T cells are exquisitely sensitive, and patients have died as a result of receiving T cells that can recognize the normal organs. They're destroyed just as easily as a tumor is destroyed. And so these two problems, the inability to identify monoclonal antibodies that exist uniquely in cancer, and that normal cells that bear any antigen, no matter how small, will be destroyed. These CAR T cells, as well as T cell receptor cells, can find tiny deposits. Again, 1 to 5 of these peptides in the surface of a cell will cause its destruction. And most of the monoclonal antibodies that are in use today actually inhibit growth factors by reacting with growth factors on the cell surface and can inhibit growth of the cells, but rarely result in their death. And so we've performed a variety of studies with CAR T cells following our description of the value of CAR T cells for patients with lymphomas. We performed clinical trials and dose escalations in patients targeting the anti-VEGF receptor that had increased expression on tumor vasculature. We saw no responses. We've treated 18 patients with an anti-EGFR variant 3. You've just heard about that. In escalating doses, we went up to 6 times 10 to the 10 cells that we administered far greater than have been given by any other group. We've never seen a response. There is one patient still alive with a fair amount of disability at 5 years, but there were no objective responses. And we've treated 15 patients with anti-mesothelin. You'll probably hear some spin about it being effective. It is not effective. We've never seen a single response to anti-mesothelin CAR. So I'm not familiar with any CAR that is effective in adults with any solid cancers, and we therefore need to look for methods to use this kind of cell transfer therapy for these over 500,000 people that die each year of this problem. Well, I'm going to talk about common epithelial cancers, but most of our experience started in patients with melanoma, and I'm just for a few minutes going to spend some time on that experience because it presented us the principles that we might apply to the treatment of common epithelial cancers. Now, in melanoma, that has so many mutations, it's the only tumor in which you can take a melanoma, make a single cell suspension from that tumor, grow out the lymphocytes in interleukin-2, which will outgrow all other cells. You can end up with pure populations of lymphocytes in anywhere from 3 to 5 weeks, and transfer them to patients in whom you've performed a lymphodepletion, a transient lymphodepletion that recovers normally within about 8 days. Well, you can see here in 194 patients that we've treated with widely metastatic melanoma. 46 of these patients had a complete response, and you see on your right the duration of these responses in months, and I'd point out two things. First, only 2 patients that have ever had a complete response have ever recurred, and that's with a median potential follow-up now of over 7 years. There's no relationship between the bulk of tumor and the likelihood of having a complete response. And so this idea that treating minimal disease is going to be more effective than gross disease doesn't appear to be the case for immunotherapy. If you have an antigen and that sets off an alarm bell in a T cell that then stops at that location, gets activated, divides, they can divide 1,000-fold in a week, 10,000-fold in 10 days. And so if you can induce complete responses, they're almost always durable. The second is that of these 46 complete responses, only 2 patients received a second treatment. So 44 of these completely responding patients received just 1 dose of T cells. And this is a living treatment. The cells go in, they're alive. We've shown that they can expand over 1,000-fold in the week after they've been given. They infiltrate into tumors and by a variety of mechanisms can result in their destruction. And these cells appear to survive as long as that patient survives. They drop in percent and frequency, but they're there for as long as we try to measure them using sensitive techniques. Of course, you don't need them there after the tumor's disappeared. It's different than when you're trying to treat or prevent infectious diseases. If you can get the tumor to have a complete response, you no longer need those cells present. And so adoptive cell therapy in this setting appears capable of controlling the last melanoma cell, as you can see from these complete responders in the upper graph. So what do these 2 recognize in melanoma that allows the in vivo destruction of the last melanoma cell? And is that something that we can use to treat patients with the common epithelial cancers? Well, we were seeing these complete regressions in patients with bulk melanomas in the absence of any off-tumor, on-target toxicity. And that suggested to us then that in fact, perhaps it was the only absolute difference between a cancer cell and a normal cell that existed in cancer, and that is the unique mutations that turned that normal cell into a malignant cell. And so for the non-immunologists in the audience, I just want to emphasize this one point that's critical for understanding what I'm about to talk about. And that is, and you've heard it in the previous talks as well, that conventional T cell receptors that are present on cells are alpha and beta chains that recognize a peptide in the groove of an MHC molecule. And so intracellular proteins, to be recognized by conventional T cell receptors, do not recognize the 3-dimensional structure on the surface of a cell, a protein on the cell, which is what CAR T cells recognize. They recognize this small peptide, anywhere from 8 to 12 amino acids, that are in the groove of that particular patient's MHC molecule. And that's true for both CD8 and CD4 cells. And so how do we study what the T cells that recognize something on malignant melanomas and resulted in their what appear to be curative destruction? So we've developed this technique that I'll show you that we just reported a few years ago and have been refining and I believe improving to attempt to generate T cells that recognize the unique mutations, the mutations in any patient's cancer. And so this is how we do this. We isolate genomic DNA and RNA from a tumor deposit. We do a whole exome and transcriptome sequencing to identify all the mutations. We sequence the tumor as well as normal cells. That can be done in any laboratory with NextSeq Illumina apparatus in 8 to 10 days. You then know every mutation that's present in that cancer. And they vary in melanoma up to 2 to 3 to 400 per tumor, and most of the epithelial cancers between about 50 and 150 mutations. Well, how do we find out which mutations are recognized by the T cells that mediated complete regression? Well, the key to doing this is to synthesize 25-mer amino acid, either polypeptides or mini genes that encode 25-mer bases that again encode this 25-mer peptide. And so the key, as you can see in this top 25-mer here up in the left of this slide, we've put together 6 different individual mini genes, or again 25-mer peptides in a string. We call this a tandem mini gene. We then produce RNA by in vitro translation from this mini gene and put it into a patient's own antigen presenting cells that present all of the class 1 and class 2 molecules of that patient. We then take this antigen presenting cell that has all of the patient's class 1 and class 2 molecules and into which we've put all of the mutations, that is genes that encode the mutations or peptides, 25-mers, that we can pulse onto the antigen presenting cell. And because it's a 25-mer with the mutation in the middle, that 25-mer has to encode every 8 to 12 amino acid peptide that could possibly be recognized by a T cell. And so the advantages of this approach are, one, you don't do any predictions of what is a mutation. You measure them. There's no need to, what we do as well is we test in one assay all candidate peptides and all of the MHC molecules of that patient, because of course the peptide has to fit uniquely in that MHC molecule. And you don't need a tumor cell line in which to test specificity, because by combining all the 25-mers that encode every mutation, you have an avatar of the tumor cell. And one of the things that's promoted immunology research in melanomas is it's easy to grow those in culture, but as you know, it's very difficult and often not possible using current techniques to grow tumor cell lines from the common epithelial cancers. Well, you don't need them to do this particular assay. Well, we started this work in 22 patients with melanoma. They had a median of 318 mutations, total of over 13,000. We used RNA-seq to see which were expressed. We tested all 4,000 of these mutations, can identify 54 immunogenic epitopes. We have not published this yet. Patients with mutation-reactive cells, there were 18 of the 22 we looked at. And interestingly, virtually all of the mutation reactivity in the T cells were CD8 positive T cells. When we looked at the antigens that were being recognized, because we could take these 25-mers and then deconvolute exactly which of the individual antigens in the mini genes were being recognized by patients that had a complete regression. Every patient was recognizing an absolutely unique mutation. There was no sharing of any mutation in 2 patients. Of these 54 immunogenic neoepitopes, none were shared at all. Every patient recognized something unique in their cancer. And many of these patients recognized many. One patient recognized 10 of these mutations. Well, we've now done this very carefully in 72 patients with gastrointestinal cancers with highly vetted data. These patients, these 72 patients, had a total of 10,261 mutations. About 7,400 were actually expressed by the tumor by RNA-seq measurements, so we could screen all 7,496 genes. 120 of them turned out to be immunogenic. That is, encoded a protein product on the surface of the cell that could be recognized by that patient's own T cells. And so there was the possibility that just as in melanoma, all of the complete responders were recognizing unique mutations, would it be possible to identify T cells that recognize these unique mutations in these patients? Now, half of these were CD8 and half were CD4 in the epithelial cancers, in contrast to melanoma. And once again, somewhat shockingly, of the 120 immunogenic antigens that gave rise to T cells, 119 were absolutely unique to that individual patient. Only 2 patients shared a KRAS mutation that was restricted by CW0802. We've now extended that beyond the GI cancers to patients with breast, lung cancer, ovarian cancer. And you can see, if you run down that third column, you can see that in virtually every epithelial malignancy, about 80% of patients with the techniques that we originally developed, and I think we're getting better at it, have T cells that can recognize the individual mutations in their cancer. So there's no such thing as an immunogenic tumor and a non-immunogenic tumor. It's just melanomas are unique because they have so many mutations. You generate a lot of T cells against them. Unselected TIL can just cause regressions. For all of the common epithelial cancers, these cells are very rare, but you can find them. And 80% or so of all patients with these solid tumors do contain these T cells. And in fact, of these 197 antigens recognized by T cells, 196 are unique. Only 2 share the same exact mutation. And so we're developing techniques now that are quite complex, and we're struggling with this, to identify the exact small population of T cells that can recognize the cancer. But in fact, in about 45 patients, in 15%, we've been able to identify T cells that recognize mutations that can cause the regression by RECIST criteria of these metastatic solid tumors refractory to all existing treatments. The first patient we treated was this young woman who had had a cholangiocarcinoma. This is a bile duct tumor. She had had half her liver removed and had a fairly typical story. She developed lung and liver metastases. She received chemotherapy, didn't work. We gave her unselected TIL, it didn't work. We then developed a technique in which we could specifically identify the unique cells recognizing her mutation. There are a variety of ways to do that, by getting multiple clones, starting with tiny numbers of cells, or looking at 4-1BB upregulation when the cells are stimulated with their antigen. We gave her her own cells that targeted one of the 26 mutations that she had. It turned out the mutation was against an ERB2 IP, single non-synonymous mutation, and we could identify it and isolate it by FACS, as you can see circled in that red square. And here's an X-ray of the patient on the left, and with what she started with. You can see those large tumors, each pointed to by a yellow arrow. All of these tumors went on to disappear, with except for 2 lesions. She also incidentally had 3 liver lesions, which went on to disappear, regress completely. She was left with these 2 deposits that you can see on the left. Because we had increased the precursor frequency so dramatically, we gave her pembrolizumab, and in fact those lesions, that top lesion disappeared, the bottom lesion shrank. It's now barely PET positive, has not changed for the last 2.5 years, and this patient is now well over 5 years later living normally, I think with no tumor, with the exception of that one scar on the left. Because this is a treatment approach that's not unique to any individual cancer, it's targeting mutations, and so potentially eligible for a variety of cancer types, and we've seen and reported responses not only in that cholangiocarcinoma patient, but in patients with colon cancer, with cervical cancer, and this patient with breast cancer that we just reported a few months ago, who again had a fairly typical history of having gone through multiple different chemotherapy and hormonal regimens, came to us to receive her own cells that recognized her mutations. Interestingly, in this patient, we could identify 4 non-synonymous mutations in 4 fairly random proteins. It's these random somatic mutations, which are the bad luck mutations that cause cancer. None of these are driver mutations. But by targeting multiple at the same time, we could overcome the heterogeneity that existed in cells. And this patient, who was treated in December of 2015, underwent a complete regression of all of her cancer and is still now completely disease-free 3 years later. You can see the tumor on the left anteriorly is starting to grow through the chest wall, multiple liver metastases, all of which disappeared, and she is in an ongoing complete response. So one hypothesis based on these results is that it's the recognition of random somatic mutations which is the final common pathway that explains cancer aggression for virtually all of the natural immunotherapies. It's been shown by Ton Schumacher now that anti-CTLA-4 treated patients can recognize mutations. Anti-PD-1 checkpoint modulators are most effective in patients with large numbers of mutations, melanoma, smoking-induced lung cancers, and certainly TIL. The second, which is a somewhat disturbing conclusion, is that any intracellular protein can potentially be a cancer antigen if it's mutated, processed intracellularly to a peptide, and then bind to one of that patient's MHC molecules. And so what's in tumor antigen in one patient, because they have the right MHC molecule to bind to, that same mutation would not be antigenic in another patient who had different MHC molecules. So there's good news and bad news. The bad news is that treatments targeting these mutations are going to be highly individualized, because you're going to have to develop basically a new drug for every patient, as we do with CAR T cells, but not against shared antigens, against the unique cancer antigen of that patient. The good news is that because all patients have mutations, there's at least a possibility that a wide variety of different cancer types can be utilized for this kind of therapy. Well, there are potential improvements that we're looking at quite strongly. One, to find the T cell receptors and put them into PBL of patients, to sort the mutation-specific cells. We reported that PD-1 is a good marker for the tumor-infiltrating lymphocytes that have anti-tumor activity, and a variety of other potential improvements. Well, I'll finish with this very brief discussion of mutations in driver oncogenes or tumor suppressor genes that are indeed shared among patients, except it's very uncommon that you can actually find them in an individual patient that will match the patient's MHC molecule. KRAS is one. We've identified, as I mentioned, these 2 KRAS receptors as part of our screen, but using in vitro sensitization techniques, we've been able to find others, and we're beginning to accumulate a library of T cells that can recognize KRAS mutations that are very common, 40% of all cancers, 70% of pancreatic cancer patients. But notice we have to pay attention to the MHC molecules of that patient, because even though you have one of these common hotspot KRAS mutations, you also have to have the appropriate MHC molecule. We're also generating libraries against p53 mutations. Again, 50% of all cancers will have a p53 mutation, but again, you have to match it with the patient's own MHC molecules. And so if we can generate a large enough library, then we might be able to have actually off-the-shelf reagents for some selected patients that have KRAS mutations and p53 mutations with the right MHC restriction element. And so I might leave you with a potential blueprint for treating common epithelial cancers, and that is to target the immunogenic somatic mutations that are unique to that patient's autologous cancer, or, as we mentioned just a moment ago, raise a library of T cell receptors against the rare shared cancer mutations. And so it appears as if cell transfer therapy can mediate durable regressions in patients with metastatic cancer refractory to other treatments. The T cells that recognize unique somatic mutations can be found in TIL, and we've also reported now finding them in peripheral blood at about 100th the frequency in patients with common epithelial cancers. And that identification and targeting of these mutations unique to each cancer or shared mutations such as KRAS or P53 have the potential to extend cell therapy to patients with common epithelial cancers. Well, thank you for your very kind attention.
Okay, time for one question.
Nice talk, sir. My question, I'm sorry.
We can give you a chance for a question too, because it's half an hour before
do all the T cells have the specificity for all the mutations, or they develop over the period of time? And if they do, at what stage of development of cancer would you detect them?
I'm sorry, I didn't quite understand what the question was.
My question was, do all the T cells have the specificity to recognize the mutations in the tumor? And if they don't, then at what stage do they develop the specificity?
Right, so every T cell that I've mentioned, and the ones that are recognizing antigens, are recognizing unique antigens that are present on that tumor. And you can show that when we do have tumor lines, when we can test against fresh cells or tumors that are grown in xenogenic mice, or by putting the entire gene into a cell and show that antigen can be successfully processed by the tumor cell. Now, because tumors grow and can gain and lose antigens, it's important to be able very often to treat with more than one T cell receptor recognizing multiple antigens. And in fact, finding those T cell receptors in peripheral blood probably integrates the entire life of that tumor as people have developed immune reactions to antigens that are gained and lost. And so we're working quite hard and have published our initial efforts to identify these cells in peripheral blood that might be used for therapy as well.
Okay, last question. Thank you.
So you make a very compelling case that patients' TCRs are really directed against neoantigens, mutated proteins. Right. Your mini-gene approach is specifically, though, focused on those mutated proteins. So my question is, do you think that any aspect of a patient's T-cell response to their cancer is directed at non-mutated proteins at all, or is it all directed at these mutated neoantigens?
You can find T-cell receptors against some differentiation antigens, most famously melanoma, where you use T-cells and you can get some vitiligo. But when we did gene therapy studies by finding high affinity T-cell receptors against these differentiation antigens like MART1 and GP100, and use those gene-modified cells to treat patients, patients began to become both blind and deaf because of the MART1 expression in the eye and in the ears. Now, we could revert that back to normal in every patient by giving transtympanic injections of steroids or transocular steroids. But the number of melanocytes in the ear are tiny. They're in a tiny layer of the stria vascularis, a tiny single cell layer. T cells can find them because every cell that's alive has access to the bloodstream. So it's very dangerous. HER2, for example, which we tried to target with a lethality in a patient because it's expressed on some normal cells. Yes, it's overexpressed on some breast cancer cells, but it's also expressed in all normal cells. So you have to be extraordinarily careful not to target any target or any protein or peptide brought to the surface that's also expressed on a normal cell.
And cancer testis antigens that are presumably not expressed in normal
We've studied
tissue?
them extensively. I think if you have synovial cell sarcoma, where you have a mutation, a translocation in the X chromosome that leads to very high expression of NY-ESO-1, you can see some regressions. But we've just published over 4,000, a survey of over 4,000 patients. It was published by Sid Kirkar with a variety of epithelial malignancies. And low single digits of those common cancers will express NY-ESO-1 homogeneously. And so Because it's 2-3%, if you're going to use those cancer germline antigens, you're going to have to screen 100 patients to find 1 or 2 to successfully treat.
Thank you.
Well, thank you.
All right, well, thank you very much for your attendance at the session.